So in this recording, we want to address our next biomacromolecule, proteins. Now remember, we've got four classes of biomacromolecules: lipids, proteins, carbohydrates, and nucleic acids. We've talked about lipids. Now, proteins, carbohydrates, and nucleic acids can all be categorized slightly differently from lipids in that they are polymers, where lipids are not true polymers. And what we're going to see is that there's some commonality to the chemistry of life. This provides efficiency for life. And this, in the simplest form, is known as a polymer-based approach to life. Right, this is our first example of kind of this chemical efficiency. So first we need to define polymers. So polymers are large molecules composed of smaller organic molecules linked together. And we're almost always going to visualize them in these cartoons as kind of like beads on a string. So the monomer is just some small organic molecule. And we link them together and to make these functional larger molecules, right? Now the cool thing about this idea of a polymer approach to life is that the chemistry that links the monomers and builds polymers is the same for all three classes. So we have what's called a dehydration synthesis, this dehydration reaction, where we can see the short polymer 1, 2, 3 linking the fourth one is drawing a water molecule out from the process. Dehydration, drawing the water out. Where when we want to break a molecule down, like when you eat your food and you want to break down the protein in the meat that you've eaten or the vegetable from the vegetable that you've eaten, and use that yourself, you want to break it apart, you do what's called a hydrolysis reaction. Hydro, water, lysis, to break open. And you revert to that monomer being free from the polymer. All right? So this is an approach that we're going to see for proteins, making carbohydrates, specifically polysaccharides, and nucleic acids. Again, it's efficient because instead of having to build a large molecule atom by atom, what I can do is just have a common base, a common pool of monomers, and link them together any way I want. Just like when I go to the hardware store and I buy wood and nails and bricks and mortar, from that pile of common elements, I could build a house. And I could take it apart and build a completely different house from the same pieces. Right? And that's how life's going to work. It's much more efficient than saying every time I want to build a house, I have to fire the bricks and grow the trees and make the lumber and dig for ore and forge the nails. Right? That takes way too long. Not that life doesn't do that, but when I can, I want to kind of disassemble and reuse those parts to build something new. That's this polymer approach. All right? So I want you to really think about that and understand it as we talk about the different polymers. So again, our first polymer that we're going to talk about is proteins. And when I talk about proteins and ask you what proteins do, I want you to say that proteins do stuff. Right? So say it now. What do proteins do? They do stuff. Because proteins are going to be the workforce of life. Right? They're really what's inside the bag that is created by the cell membrane that direct the chemistry of life. They can be structural. We talk about your hair or spider silk, or we talk about many of the proteins in your muscles, right? They can be transporters and carriers. All these proteins do stuff. But most importantly, and we'll talk about this in greater detail later, is that they are enzymes. And enzymes catalyze chemical reactions, meaning they promote and direct and control chemical reactions. And that's what proteins are going to do. What do? They do stuff, right? So for example, let's go back to that cell membrane, right? So remember we talked about lipids, and this lipid bilayer makes up the cell membrane. But in reality, that lipid bilayer is full of proteins. And we oftentimes will see proteins in this kind of cartoon, this big glob, right? This like glob of stuff, right? And in fact, that's not very detailed. We're going to see that that in fact is made up of many, many amino acids all folded back on themselves to make that glob. But the proteins in the membrane are going to carry things across that we need outside and inside the cell. All right, so they're doing things. Now let's look at some proteins. So this is a depiction of the enzyme lysosome. This is stuff that you find in your tears that helps to fight bacterial infection. But ultimately what you see here is this ribbon kind of structure, and then again the space-filling structure where you can see each atom. And what we're going to learn is the shape of a protein that is ultimately important for its function. Form and function is another very, very common theme that we're going to see in biology again and again and again. So as we look at these proteins, here's collagen. This is the material in your ears and your nose that gives it flexibility and support. Hemoglobin carries oxygen in your blood. But you can see it's this twisted chain that's folded back on itself that makes up the shape of this protein. And so what makes up that chain? This is a polymer, right? Beads on a string. What are the beads? In the case of proteins, those beads are called amino acids. So proteins are often known as polypeptides because what we do is we link those amino acids together in what's called a peptide bond in the dehydration synthesis reaction. So when we look at those different amino acids, we're going to learn that basically there are 20 common amino acids that all life uses. Now we can look at those amino acids, and what's highlighted in the white square is what's called the R group of the amino acid. This is the part of the amino acid that's different for each amino acid. Now if you look at this group, you see on the far left there, they're the nonpolar amino acids. Nonpolar, just like the lipids. So all of these amino acids are not going to want to mix with water. Why? Let's look at them. Look at leucine, for example. So on the upper row, one from the right, you see leucine. And that R group is a whole lot of hydrocarbon, right? And remember, hydrocarbons are non-polar bonds. There's no polarity there. There's no charge in that R group. And so it doesn't want to see with water. It doesn't want to be with water. It's non-polar. So all of these we can lump together as the non-polar amino acids, and in that general chemical category, they're going to respond basically the same way, although each amino acid has its own characteristics. Now compare and contrast these to the polar or charged amino acids. These now are going to want to be with water. Right? So, and they're different sizes. Some are big and some are small, and some have like a full charge on them, like lysine or arginine, right? Or glutamic acid or aspartic acid. And they're going to really want to interact with water and can do other kinds of cool chemical things, right? And that's the thing. Each amino acid has its own unique chemical characteristics. And by combining them in specific combinations, we get proteins with very unique characteristics. So what we're going to do is link those individual amino acids with a, boom, there it is, dehydration synthesis. See that top part there? Forms what we call a special peptide bond. And then we start to build this amino acid. It has a backbone with those R groups, those chemically different groups hanging off the side that are going to build the different chemical characteristics of a protein. Right? So proteins, again, are made of amino acids, and you want to think of each amino acid as kind of like a train car. And when you put them all together, you get a train. Right? Now, the cool thing about that is that each train car has different specific functions. Right? So if you want to build a train to move people from Seattle to San Francisco, make a train with an engine, that's amino acid 1, and then a bunch of passenger cars. Right? That could be lysine, 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 all a particular kind of amino acid, and then maybe a caboose. I don't even know if they use cabooses anymore. And that is a protein. Now, you can make a very different kind of train to move people and cattle and cars. Those are going to be different kinds of cars. They're different amino acids that you link together. Still a protein, but a very different function. Right? So that's kind of the train analogy for how proteins work. Now, as we explore proteins, what we can get into, and I need you to understand, is protein structure. And there's going to be 4 levels of protein structure that we need to look at and kind of get down in our head. Because by understanding protein structure, we're going to ultimately understand how genes work. All right? And that will come later. But for now, we're going to walk through these 4 levels. The first level is called the primary structure. And that is simply the sequence of amino acids. So in this case, we can see starting at the top, amino acid 1 is LYS, that's lysine, followed by valine. Phenylalanine, glycine, arginine, cysteine, glutamine, leucine, alanine, right? 1 through 10 there. So simply writing on a list in order those amino acids, that's the primary structure. And depicted here, you see them just like beads on a string. All right? That's the very first level. And that's how we're going to build a protein, by linking those amino acids in that order. Now, because each of those amino acids has its own unique chemical characteristic, some wanting to interact with water, some not wanting to interact with water, and differences in between, you see them start to fold. And so what we talk about is a protein becoming natured or denatured. Right? A normal protein is going to fold up into a very specific shape based on the chemistry of the amino acids in the order of its primary structure. When we unravel it, we just get beads on a chain, and it doesn't do anything. But when it forms a specific shape, it now is a tool for chemistry in the cell. Right? It's like a hammer. Like a hammer is the shape that it is because it's built well for driving nails. I wouldn't try to use a fork to drive a nail into the wall. It's not the right shape. It's not built the right way, form and function. Right, so the primary structure of protein is the order of amino acids. Now, let's look a little bit closer at this folding process. So secondary structure then is what we call microstructure. And so the secondary structure is coiling of those amino acids into what are called alpha helices or beta-pleated sheets. Or if it doesn't fall into either of those two categories, we just call it random coil. So secondary structure is alpha helices, beta-pleated sheets, and random coil. And these form immediately, right, based on the local chemistry of the different amino acids in the order that they come in on the polypeptide chain. So the alpha helix is just that. It's a helix. Beta-pleated sheet is back and forth. It makes these sheets of amino acids. And that is secondary structure. Secondary structure then packs together to form the glob that we've seen as these big purple balls. This is called tertiary structure. So on this slide, we can see all four levels of protein structure. Primary structure, valine, glycine, serine, leucine, the order of those amino acids. To the secondary structure, alpha helices and beta-pleated sheets, that then pack together to make the shape of the protein. And that is what we call tertiary or third level structure. Now, some proteins will actually have what's called a quaternary structure. Not all do, but some. Quaternary structure is then when you have many proteins that pack together into a larger complex to make that complex functional. All right? So primary structure, secondary, tertiary, quaternary. You need to know what each one of those are. Now, to talk a little bit more about form and function, let's take an example of how altering the primary structure of a protein can affect all the subsequent levels. So in this case, we're looking at, and we'll come back to this slide again later when we talk about genetics, but this is a single amino acid substitution in a protein, hemoglobin, that causes sickle cell disease or sickle cell syndrome. So in this case, if you look at proteins 1, 2, 3, 4, 5, 6, in a normal red blood cell, the hemoglobin at position 6 is a glutamine. It has an amino acid called glutamine. If you have the genetic disease sickle cell anemia, then in that 6th position, instead of glutamine, you have valine. And that valine happens to be in a very, very important position. Because when that valine appears in position 6, it changes the shape of the protein such that its whole shape changes and it starts to glom together into these long chains of proteins that sickle the cell, that make it this long sickle-shaped cell. And that's not a very good cell for carrying oxygen. All right? So literally here you can see the round red blood cells in this microscope slide and the long thin ones that are sickled. Okay, and that can jam in capillary tubes and cause great pain and lead to the many symptoms of this disease. So again, we want to think about this idea of proteins being natured or denatured, right? So we've got a protein that is folded correctly, it's natured, versus folded incorrectly, denatured. All right? A protein is called active or functional when it's folded correctly versus inactive or killed. Right? We talk about killing a protein. How do you do that? You can do it with heat. That's why we cook things. When you crack an egg into a hot frying pan, you see that clear white of the egg go white and solid. That's actually proteins, albumin being denatured and glomming together to form that solid. Heat is denaturing the protein. Chemicals, salt, pH, all those things can affect a protein structure. And when it doesn't have the right shape, it doesn't work. All right, let's keep that in mind.